Hey, hi guys. My name is Allison Campbell. I am a uh, fourth year chemical engineering student. I also have the biomedical emphasis and the bioengineering certificate. So that's kind of the area that I'm interested in. Today, I'm going to be presenting with you some research that I actually did my freshman year. So I started out research in a pharmacology and toxicology lab. So I wasn't in the chemical engineering department or even the engineering department. And I really liked that just because I felt like it kind of got out of engineering so I could see sort of the applications a little bit better. And you can also see just how diverse chemical engineering is and that you can really apply that to so many different areas. And also just kind of through my experience, I really learned that no lab is ever going to be like, no, you're a chemical engineer, we don't want you. So you don't ever have to feel like trapped by your chemical engineering degree. You really can go into a lot of different things outside of that. So I was in the memory lab and the principal investigator, the PI, was Jay Subramanian. And he was actually a brand new professor. He was so kind, so awesome. But he was there, I think he had gotten there like a semester before I joined the lab. And so it was actually just him and his graduate student, Shreya, and me. And so that was really nice because I got to do everything that Shreya, the graduate student, was doing because we were kind of all in the same boat of not really knowing what was going on. Um, and so that really kind of gave me a feel for what a smaller lab is like, because you definitely can get into some of these bigger labs with a bunch of undergraduate students and a bunch of graduate students. And those are nice because you get a lot of mentorship opportunities. But then in the smaller labs, you kind of can maybe be a little bit more hands-on in some applications, which is nice. So just a brief overview of this lab. So like I said, I started this in my second semester as a freshman. I wanted to just kind of take that first semester to get a feel for college. I was like, there's no rush to it. I knew I was interested in research, but I was like, let's go in slow so I don't stress myself out. And we researched how memories are acquired, stored, and lost in the brain under normal and pathological conditions. So we kind of approached these at a bunch of different levels. So we looked at the entire brain and we studied the circuit reorganization, but then we also studied like single neurons to sort of look at these synaptic neurons. And so basically I did a lot of imaging under the microscope with these neurons, but then once the mice had passed on, we did actually go ahead and kind of autopsy them, and we would take their brain and slice it into these little sections so that we could actually look at these cross sections of the brain, what happened during their life as well. So if you look kind of over in this image to the left, I've highlighted this portion in yellow. And so currently in this lab, they use state-of-the-art multiphoton in vivo like imaging approaches. And so, but when I was researching with them, we only use just this really basic like lab microscope that you're going to use in like your bio 150 classes. You possibly even use them in high school in your chemistry classes and biology classes. And so that was nice because I was really familiar with it. But I was sad that I didn't get trained on this machine, and they give such nice like image qualities. So if you look right there, that's actually a neuron, which is something that's like what we were looking at. But when I was looking at it with a microscope, it was actually just this little itty bitty red dot that I'll show you a little bit later on. But yeah, this is an awesome diagram too from them. I literally pulled this from a publication that they have. You can check that out online if you're interested in this. I feel like it's a pretty good job of being kind of self-explanatory. Even if you don't really know what's going on, he wrote it in a way that I feel like is pretty easy to understand. So I was in charge of determining the changes to functional connectivity between brain regions associated with memory storage and how excitatory synapses are lost in mouse models of Alzheimer's disease. And so I apologize for this picture. It's a little bit grainy. I actually just took this on Snapchat when I was in the lab. But you can see this mouse here was one of our genetically modified mice. And what we would do is we would take out a portion of their brain, a little circular region, sorry, not of their brain, of their skull. We would take out a portion of their skull, and then we put a glass plate over it. And these mice were fluorescently marked in the womb, actually. And what that would do is we would put this tracker inside of it, it would circulate through their system, and it would attach to the neurons in their brain and fluorescently mark them. So then when I put them underneath the microscope and shone a specific light on them, I was actually able to see where these neurons are in the brain and see, if they died off, if more were growing, then this tracker would attach onto them so that we could see them. And that's kind of how I was able to track that. And so here you can actually see, these are like the images that I saw. So not as bougie, but you know, it's definitely still worked for what it was. So one of the cool projects that I worked on is I developed a new protocol for the neuron mapping I was doing with the microscopes using Adobe Illustrator. And so Adobe Illustrator is just something that I had previous knowledge on in high school. And we were really struggling because we had these little itty bitty brain images in the bottom left here. Those are basically what I was taking the individual pictures of. And then we didn't know how to put those together in a way that was aesthetically made sense so that we could actually see the whole image all together. And I was like, hey, you know, I can put this all into Adobe Illustrator and I can kind of map these things following the main blood vessel trees. And then once I put all those together, I can overlay them onto a big image that we took of the brain so that you can actually see where those neurons are. And so that's how I was able then to mark these little red dots that you can see in the picture above it. And those are like were the neurons that were showing up, you know, within that individual memory exercise. And then I would do that, you know, we'd have one memory exercise the day after, and then we do it, you know, 2 days after, and then a month after, and then, you know, 2 months after. And so I was able to track these things over time. And then it was cool, like I said, since it was just me and the graduate student at the time, I also got to see the training of the new graduate students during my sophomore year. So that was just kind of a cool brag because I was just a sophomore and I was like, oh yeah, come here, graduate students. I can tell you everything that you need to know. And that was just another cool perk of being in a pretty small lab and a brand new lab. So just some final notes. Research at KU is extremely engaging and it really is easy to get into. I'm sure that you probably have heard it a couple times already, but all you have to do is just send an email to the professor. That was really all that I did. And he, you know, immediately responded back and they were always really excited. And I will just say that professors are very, very busy. And so if you're, you know, sent an email and they don't respond back, don't ever take that to mean, you know, that they don't want you or that they're like, oh no, we don't like her or him. It's like, we don't want you in our lab. It's probably just because they looked at it, were really excited and then forgot to respond. And so definitely don't be afraid to send a follow-up email. They really do want you in your lab and they'll let you know, you know, if their lab is full, they'll be like, hey, you know what, we're full right now. We're so sorry, but like check back in next semester. And, you know, people will have graduated, people have moved on. So it's really just something that you kind of have to do on your own, but they really do want you in your lab. And it's also highly recommended for anyone, not just students wanting to go to graduate school. So I personally do want to go to graduate school and that's fantastic experience for that. But I have a lot of friends who are doing research right now and, you know, they're getting paid, they're taking it for an elective credit. And they're also, it's just something that they can talk about in interviews and be like, yeah, I, you know, got to run this project within this lab. And that still shows those leadership skills and team collaboration skills that are so important in engineering. So yeah, it was great talking to you guys. And, you know, thank you for listening.